Hello, and welcome to Journal Club. Today, we'll be talking about this paper from a team of researchers at the Beijing Institute of Technology. In this paper, the regulation of the enzyme alcohol dehydrogenase is examined. Specifically, this team looked at the effects of high versus low concentration cofactor NADH on the efficiency of catalyzing the reduction of formaldehyde and methanol. Following me so far? Okay, let me break that down for you. This work has a lot to do with oxidation chemistry, and in this context, oxidation basically means the addition of oxygen. So if you start with methane, oxidation goes this way, and reduction goes that way. Biochemical systems walk through this pathway with a different enzyme for each step, regulating whether the system walks forward or backwards. The reaction we care about is this one right here, and it's catalyzed by alcohol dehydrogenase. Note that for every redox reaction, something must be oxidized, something else must be reduced. Many biochemical reactions involving reduction or oxidation use the cofactor NADH to facilitate this. For the reduction of formaldehyde to methanol, NADH is oxidized to NAD+. And this is important to note here. Alcohol dehydrogenase is a bi-substrate enzyme. It uses both formaldehyde and NADH to produce the products NAD+ and methanol. This is where we get back to the objective of our paper today. The research team noticed that there was a plethora of research for the oxidation step, but less was known about how alcohol dehydrogenase reduces formaldehyde to methanol. But why does this even matter? Well, the team illustrate that ADH is the final step of the conversion of CO2 to methanol. Understanding this step would be extremely useful for taking the greenhouse gas CO2 out of the atmosphere and converting it to a fuel, methanol. So, how did they study alcohol dehydrogenase? It's actually a stupid simple procedure. They used a shaker set to this speed with the formaldehyde and NADH at specific concentrations. The reaction was started by adding the ADH, and a sample was taken after 3 minutes and put in a gas chromatography machine. Rate is just a change in concentration over time, so they were able to measure the rate of this reaction using just a shaker and a GC. Okay, but if you've ever done kinetic experiments, you'll know that the real kicker is in deciding how much concentrations of everything to start with. And that depends on what aspect of the experiment you're looking at. So the Beijing team actually did this experiment in 2 different parts. First, they kept the NADH concentration constant and varied the amount of formaldehyde in there. Then, they kept the formaldehyde concentration constant and varied the amount of NADH in there. In enzyme kinetics, we use a model called Michaelis-Menten. Other videos around the web can break it down further, but here's what you need to know. Directly plotting the rate velocity of a reaction with the varying substrate concentration gives a curve that looks like this. This curve makes sense logically. The more substrate that's added, the faster the reaction involving that substrate will progress. 2 factors are of particular importance here: the maximum velocity, Vmax, and the factor Km. Vmax is easy, it's just, the maximum velocity. Km is a little weirder though. My best sum up is that it is the substrate concentration at which the rate is half of Vmax. It is a mathematical consequence of Michaelis-Menten kinetics, and it sort of tells you how much affinity an enzyme has for its substrate. What you need to know is that a smaller Km means higher affinity. There's a third factor that pops out of here: catalytic efficiency, which in this context is Vmax over Km. If you take the reciprocal of both velocity and substrate, you get this nice straight line, in which you can easily find Vmax and Km. Back to the team. When they went from 100 millimolar NADH to 10 millimolar NADH, the Vmax decreased and the Km decreased, which means that the affinity towards formaldehyde increased. Interestingly, catalytic efficiency stayed the same. This trend is explored more when they reversed the experiment. They used high amounts of formaldehyde and varied the amount of NADH, and things got really weird. So here's the super nice looking data for the first experiment, and here's the data, for the second experiment? What? This is supposed to be a straight line. It turns out that at high concentrations of NADH, it's acting as an inhibitor. The cofactor that's needed for this reaction is slowing it down. The Beijing team call this an uncompetitive inhibitor. Wait, what does that mean? According to my enzymology class so far, there are 3 types of inhibitors. There's competitive, non-competitive, and uncompetitive. Competitive inhibitors look like the normal substrate and trick the enzyme into binding to it and not doing anything. Non-competitive doesn't affect the binding of the enzyme to the substrate, but it still stops the reaction anyway. Uncompetitive inhibitors, the type shown here, bind only to the enzyme-substrate complex to stop the reaction. Knowing that NADH is an uncompetitive inhibitor The Beijing team proposed this mechanism. Alcohol dehydrogenase first binds to formaldehyde, then to NADH. Both are turned over to their products. NAD+ leaves enzyme and methanol behind, and then the methanol leaves the enzyme ready to catalyze a new reaction. However, at high concentrations, NADH can bind to the enzyme methanol complex and stall out the reaction. Inhibition. Okay, we unpacked a lot of things here. The Beijing team at the Institute of Technology, they found that if you're attempting to use this enzyme to help scrub CO2 from the atmosphere, you should not increase the NADH levels to as high as they can go. It's going to inhibit it. Instead, you should have the NADH concentration somewhere in this regime. So I guess it's true what they say. There is such a thing as too much of a good thing. Thanks for watching. I started this series for a few reasons. For one thing, I noticed that very few people, not even very many people in chemistry, really have any idea what's up in my corner of biochemistry. As science becomes more and more niche, it becomes harder and harder to convince the public that yes, we do need funding for this very specific little project here. My hope is that videos like this will encourage the public to see research as something important to fund, and that the vast polyhedron that is the scientific community will remember to take a step back and meet the other corners and niches of their field.